Thank you, Miriam. I'll share my screen. Okay, hopefully that works fine. Yeah, thanks for hosting me and the very nice introduction, Miriam, and thanks to Nima for inviting me. I've been to a few Weekend seminars before, and it's all a bit intimidating, really. But you know, when Nima asked me, he said, you know, it's not supposed to be a very long or very serious talk. And then when I said yes, he was like, you know, it's 45 to 50 minutes. So now I'm not so sure anymore, but as a reward, he'll get a very long and very serious talk. So anyway, as you can read, the title of my talk is A Computational Perspective on NMDA Receptor Antagonism in Psychiatry. And I like this title because it sounds very broad, more like a broad research theme rather than a collection of a few studies. And it's also more broad maybe than what you'll get. So when I say computational, for example, I mostly mean Bayesian models of belief updating. And when I say psychiatry, I mostly mean psychosis and maybe a bit of depression. And when I say NMDA receptor antagonism, I basically mean ketamine. So what's with ketamine? So what is slightly mysterious about this drug is that it seems to play two parallel lives in psychiatry. On the one hand, we use it to understand the emergence of psychosis. So when healthy participants are under ketamine, they report strange perception, dissociative experiences, delusional thinking, and other features of psychosis. So they feel slightly psychotic. And we know that ketamine blocks NMDA receptors in the brain, and that this is somehow responsible for these psychotomimetic effects. And NMDA receptor dysfunction or dysregulation has also been implicated in the pathophysiology of schizophrenia. So all of that together has led to ketamine being used as a pharmacological model of psychosis, on the one hand, in psychiatric research. On the other hand, if you give the same dose of ketamine to patients with depression, in particular treatment-resistant depression, then they report feeling better within hours after taking that drug. So it's also known as a rapid-acting antidepressant in psychiatry, and the mechanisms that underlie these effects are much less established, and there's a lot of open questions surrounding this. For example, we don't know whether these two effects of the drug, the antidepressant and psychotomimetic, are related in any way. If you're interested in kind of the latest news and debates on the antidepressant effects of ketamine, then I suggest you come along to the Ketamine conference, which will be taking place here at Oxford in this April. But I think overall, this is a very good example of some of the challenges that we face in psychiatry. Namely, there seems to be an explanatory gap between the levels at which we describe the effects of drugs and the levels at which we describe symptoms and psychiatric diseases. So when we define mental illnesses and we talk about symptoms and we measure them, we're on the level of cognition, behavior, mood, subjective experience, etc. But when we treat patients with drugs, then we suddenly mess with their neurochemistry. And as you can see from the example of ketamine, we're not very good at predicting the effects on one level using the effects on the other level. So usually the way that it goes is we discover that some drug has some interesting effects, for example, antidepressant effects. And then we look at, well, what does that drug do on a molecular level? Oh, it messes with the glutamate system. So maybe glutamate might be involved in depression. And obviously we'd like to have that the other way around. So in computational psychiatry, our hope is that we can provide a missing link between the levels by describing what's going on in the brain on this more algorithmic or computational level, and basically describing the neural computations that neural circuits engage in and that we think are driving behavior. And if we can understand what drugs are doing on this level, then we might be able to better predict the effects on symptoms as well. And in my work, I've mostly been using hierarchical Bayesian models of perception and belief updating, where we think about how to optimally combine prior knowledge with new information to form beliefs. And why would you use these models for everything that you're doing? Well, I think there's probably not much left of cognition, behavior, etc., that has not been attempted to be described by Bayesian models. And wouldn't it be nice if there was just one equation that would solve all the mysteries of the brain? That would be amazing. So today I will present some of my work in 3 parts. So in part 1, I will talk about some of the things I did in my PhD, where I used mismatch negativity paradigms to look at NMDA receptor antagonism and its kind of computational function. And then in part 2, I'll talk about what I'm doing here at Oxford with Ron's group. That will be perceptual decision-making and new paradigms for measuring evidence integration and EEG. And then there'll be 1 or 2 slides on kind of future plans, what we're hoping to do with these paradigms and ketamine studies, etc. So let me start with the first part, which will be all about NMDA receptors and volatility in statistical learning. And as I said, I've been using mismatch negativity paradigms to look at this. And whenever I present this work to an audience, which is not the audience at the MMN conference, because there is a conference on MMN, then I feel I need to pitch the MMN paradigm a little bit. Admittedly, it's not the most exciting paradigm in cognitive neuroscience that you could think of, but let me tell you why it's really great anyway. So it is very simple. The way it works is you basically play a sequence of tones to participants, and the sequence has some regularity in it. In the simplest case, this would just be repeating the same tone over and over again. And then you, at some point, introduce a tone that breaks this regularity or violates the rule. In this simple case, it would just be a tone of a different pitch that you play after repeating the other tone. And that's basically it. Participants just have to listen to that sequence. They don't have to do anything. They don't even have to pay attention. In fact, they can watch a silent movie while they're listening, or do some boring visual abstraction task, which is what we usually do with them. And you will always get this signal. And what you will see is that the neural response that you measure using, for example, EEG, is different for the tones that are predictable or follow the rule, that we call the standard tones, than it is for the tones that break the rule, that we call the deviants. And in particular, you will see this very stereotyped negativity in response to rule-breaking tones or deviants. And this is one of the paradigms, or one of the things in neuroscience where we know it just works. This has been studied for over 40 years now, and this fun conference has been going on for over 20 years. And it has been, used in numerous applications. There's also visual MMN now, and so much sensory MMN. And of course, people have been using more complex regularities, and it still works. So that's nice. And obviously, it's the ideal candidate for studying clinical populations, because it doesn't really put any demands on participants. You can even record this while people are asleep or in babies, etc. But what is also nice is that even though this seems to be a very basic mechanism that we're tapping into here, of how the brain extracts regularities from its environment, it still is interesting from a clinical perspective. So we know, for example, that this signal depends on intact NMDA receptor signaling. We know that from animal studies, but also in humans, that when we block NMDA receptors, for example, using ketamine, that this signal is reduced. And it is similarly reduced in patients with schizophrenia. In fact, this is one of the most robust electrophysiological findings in this population, with very high effect sizes. Some people have also reported that the reduction of MMN under ketamine even correlates with the strength of the psychotic effects that participants are experiencing under the drug. So there's something interesting going on here. Now, when we think about this in computational terms, we think about the MMN as a prediction error signal. And if we do that, then we can immediately think about how that would update our model of the acoustic environment. And we can see interesting differences. For example, if we experience this deviant or this prediction error in a context like this, then where, after each deviant, the sequence then reverts back to the old rule, then we should, register this prediction error, but we shouldn't really use it to update our model of the acoustic environment, because it's basically just noise, and it goes away after the deviant is over. On the other hand, if we usually experience deviance in such a context where every deviant also signals the onset of a new rule, then ideally, knowing that the rule is changing quite often, we would use this first prediction error already to update our model of the environment. And so basically this speaks to the distinction between whether things are changing fast or more slowly, whether the rule is stable or not. And in using Bayesian models of belief updating, we describe this as a model update using a precision-weighted prediction error. So we change our belief in proportion to this prediction error, but the prediction error is scaled by a learning rate. And in Bayesian models, this learning rate is then a function of the uncertainty. In particular, what I've just been telling you is the uncertainty around our current belief, or the precision of our current belief. If this is high, if we think the rule is really stable, and I know the rule and everything else is just noise, then I should not use that prediction error a lot. But if I think that the data that's coming in is really precise and I'm not so sure about the rule, then I should use that prediction error to update my model a lot. That's the basic logic. So the model that we applied for the first time to in the first study using MMN looked like this. This was a paradigm, a roving paradigm, where we have this second context. So we have these trains of repeating tones followed by a train of a different pitch. So in this case, every deviant does signal the onset of a new rule here. And we hypothesized that participants would be inferring on the probability of transitioning between tones of different pitch. So kind of a transition matrix or a matrix of probabilities. But on top of that, we also hypothesized that the brain would also infer on the level of stability of that rule that underlies the auditory sequence. So how fast this transition matrix might be changing. And we use that model to extract for every tone in the sequence an estimate of model-based precision-weighted prediction error on two levels. So the lower level prediction error, the green one, would serve to update the belief about the rule, about the transition matrix, and the higher level prediction error would serve to update the belief about volatility in the environment. And then we use these model-based estimates of prediction error in GLM as regressors to explain signal trial amplitudes across time and across the whole sensor space, and obviously corrected for multiple comparisons. And the study in which we first applied this model was a study in healthy participants who performed this MMN paradigm twice, once while they were under placebo and once while they were under ketamine. And what we found in the placebo condition was that the lower level prediction error did explain variance in the EEG signal around the time of the classical mismatch negativity component in the same sensors, etc., such that EEG responses were more negative for higher prediction error. So exactly the way we would expect according to the MMN. And then we found that the high level prediction error also explained variance in the EEG signal, but somewhat later. In particular, this later component between 200 and 300 milliseconds, which is more like the P3A, which is a related but separate component from the MMN itself, where higher prediction errors resulted in more positive EEG responses. And then when we compared these results to what we saw under ketamine infusion, we found that ketamine did not significantly change the representation of the lower level prediction error. There were some trends, but it wasn't significant across whole sensor space, and also not significant when we focused on a region of interest in classical MMN sensors and time windows. But we saw that ketamine did reduce the representation of the higher level prediction error, which serves to update volatility estimates. Again, this is true both across the whole sensor space, but also in this region of interest analysis, where the effect of the higher level prediction error explained the difference between ketamine and placebo, both in this early time window for the classical MMN and in the later P3A time window. We thought this is interesting. It suggests that ketamine interferes with higher level belief updating rather than lower level belief updating, something about volatility. And it relates to the literature in that there's been a few studies suggesting that patients with schizophrenia or people at risk of developing schizophrenia might also show a misestimation of volatility in the environment, in particular, an overestimation of volatility. So for the second study, I will go through the results somewhat more quickly because you know the logic now, but I will motivate the study a bit more carefully because we then asked whether the MMN can actually help us address a clinical question. So say we've now maybe found a mapping from NMDA receptors via computation to the symptoms, but what is the clinical question that we're trying to address? And I think ultimately the challenge that clinicians are facing is that they're confronted with patients who show a certain set of symptoms, maybe overlapping symptoms, but then they react very differently to the same course of treatment. And so we don't really have a good way of predicting from their symptoms what the best course of treatment would be for an individual. And the mostly used, most common treatment at the moment for schizophrenia are atypical antipsychotics, which all target dopamine D2 receptors in the brain. But interestingly, some of these antipsychotics additionally have a very high affinity towards muscarinic cholinergic receptors in the brain, in particular clozapine and olanzapine. And these drugs have generally a very different clinical profile from the other antipsychotics. We know that dopamine and acetylcholine both are potent modulators of the NMDA receptor. And so this has led to the hypothesis that maybe a dysregulation of the NMDA receptor might be something like the final common pathway for schizophrenia. But one axis which might explain heterogeneity in treatment response across patients might be whether the origin of the problem is more dopaminergic or more cholinergic one. And in fact, there's evidence for a subgroup of patients who show reduced availability of muscarinic M1 receptors in some parts of their brain. So this is would potentially be a helpful axis to distinguish. So what we were asking in this study is basically whether the MMN would be differentially sensitive to these two different neuromodulatory systems. And there's reason to believe it would. There aren't a lot of dopamine receptors in auditory cortex, but also we know from previous studies that the MMN is sensitive to nicotinic cholinergic modulations, and probably not very sensitive to dopaminergic modulations, although it wasn't particularly clear. And what hasn't been looked at so much was whether MMN is modulated by muscarinic receptor changes. So we conducted two studies with the MMN between subjects, pharmacological studies where participants could either receive placebo or in study one, amisulpride, which at the dose that we gave it would block D2 receptors, or biperidine, which is this relatively selective muscarinic M1 antagonist. So basically in study one, we were blocking dopaminergic and cholinergic receptors. And in study two, they could receive either placebo or levodopa or galantamine, both of which elevate levels of dopamine and acetylcholine respectively. And then to follow up on what we saw in the ketamine study, we used a new paradigm with just two tones, where one tone serves as the standard and the other one is the deviant, but the roles switch across the participant, and I manipulated the speed at which these switches occur. So in some phases, the underlying rule in the tone sequence was very stable, and then in other phases it was more volatile. And this allowed us to contrast MMN during stable phases to MMN during volatile phases without applying any model. And I won't go into detail about the kind of model-free ERP analysis. We described that in detail in the preprint, which you could check out. But what I really want to show here is that if we apply the same model or kind of the same idea from the ketamine study using this hierarchical Bayesian approach with a low-level prediction error and a high-level prediction error, then we find very, very similar activations. So again, the low-level precision-weighted prediction error explain variance in this classical MMN component, and the high-level prediction error explained this later component around 300 milliseconds. And this is true across all drug groups and across both studies. So that was very nice to see that this replicates very well. Now let's briefly look at the drug effects. So overall, our results were very supportive of the idea that the MMN is differentially sensitive to these two neuromodulatory systems, because the only drug that showed any effect was biparidine. So blocking M1 receptors in the brain affected both mismatch-related components, but also the interaction of mismatch and volatility. And in the model-based analysis, this translated to an effect on both the lower level and the high level prediction errors, both of which looked like a shift in the latency of the main component. Yeah, again, as I said, we found very consistent results in the model-free analysis. Surprisingly, no effect of galantamine, of elevated acetylcholine levels. And we speculate about this a bit. You could interpret the effect of biparidine as kind of reducing the tonic aspect of the learning rate. So the idea is that without biparidine in the placebo group and all the other drug groups, learning rates were tonically very high. And so the effect of environmental volatility on top of that was not as strong. But as soon as we reduced that tonic volatility or tonic learning rate using biparidine, then this induced or enhanced the interaction between volatility and mismatch in our data. But this is just speculation. We are working on approaches to directly fit our model to the EEG data, because as I've said, the MMN is a passive paradigm, so we don't really have any behavior to get single subject estimates of these parameters. And when we do that, we do find a reduction in the tonic volatility parameter in our model, which is nice, but this approach is still in its infancy. And so again, no dopaminergic effects whatsoever, which kind of supports the idea that we might be able to use the MMN to distinguish between patients who have the origin of their NMDA receptor dysregulation more in the cholinergic system. And we're testing this in an observational patient study to see whether this paradigm can predict whether someone would profit from a switch towards these muscarinic medications as opposed to the purely dopaminergic ones. Okay, and before I move to the second talk, second part of my talk, I just want to briefly slot in this. So this is a study that we've just written up and will be on bioarchive soon. It was a collaboration with Ilvana and Marta Garrido at the University of Melbourne and Queensland. And here Ilvana used a very similar auditory MMN paradigm to the one I showed you just before, where there's just two different tones and the probabilities of hearing them switch in kind of more stable phases and then more volatile phases. But she added in two things. So first of all, she turned it into an active paradigm. So now participants are supposed to indicate the proportion that they think is currently displayed in these tones, so which tone is currently presented more often. And the cover story is something about different birds sitting in a tree and participants having to estimate the proportion of the two kinds of birds given the sounds. And then she also added in cues. So before every block started, participants would see a short video clip that would give them a cue about the proportion of the different birds. And in uncued blocks, this cue would just be not informative at all. So there are a lot of interesting effects in this data set, and she also used fMRI while people did this. But basically, she's run this in healthy participants and schizophrenic patients. And what I want to show you is that if we apply the same model to these data and fit it to behavior of participants, because now we have behavior, then we see that the patient group had a lower value in the coupling parameter of our model. The coupling parameter is basically the parameter that scales the impact of the higher level belief onto belief updating on the lower level. So basically, the lower this parameter is, the less you're taking into account changes in phasic volatility to change your learning rate or your belief updating on the lower level. And we also found that this correlated with the degree to which both healthy controls and patients experience psychotic symptoms in their day-to-day life. So this is the KIT score that's displayed here. And so the more psychotic experiences you report, the smaller your coupling value was. So that was quite nice. Okay, so as a quick summary of that first part, we've seen that you can scale your prediction error using an adaptive learning rate that responds to changes in the stability in your environment. And the effect that this has basically is if you have a high learning rate, it means you're discounting previous evidence faster and you're focusing only on the most recent evidence to guide your belief. If you have a low learning rate, it means you're taking into account more evidence from the past. You can construct these integration kernels, basically, which tell you, the flatter this is, the more the further I'm looking back into the past to guide my current beliefs. And we saw that this should ideally depend on your expectations about how stable the world currently is. If you think things are changing quickly, then you should focus on more recent information and the other way around. So keep this thought in mind, because now it will switch to something seemingly very different, but we'll get back to this. Okay, so what I'm currently doing here at Oxford with Lawrence is working on novel decision-making paradigms. And in particular, Lawrence's group has developed a new perceptual decision-making paradigm. You might be familiar with the classical random dot motion task, where people stare at a display of dots moving around, and their task is to indicate whether they're moving more towards the right or more towards the left. And usually this is presented in trials. So there's a trial onset, you get the stimulus, and then it has probably a constant coherence, so a constant proportion of dots moving to the right and to the left. And you have a certain time to give your response, whether you think it's more to the left or more to the right. Now, this novel task is continuous in that participants are presented with this display of dots for 5 minutes in a row. And they're not being signaled when the trial starts, but they constantly have to observe the stimulus to detect changes in the coherence of that stimulus. And then whenever they think dots are currently moving more to the right or more to the left, they press the corresponding button. And so their choices can be either correct or in the wrong direction, or they can have false alarms, which means that the actual coherence in the stimulus was 0, but they thought it was more to the left or more to the right. That would be a false alarm, or they could miss changes in coherence. And the way that the stimulus is designed is that we have this mean coherence, the black line, which changes more slowly over time. And this is what defines kind of the trial periods or the signal change periods. When the black line is at 0, it means we're at baseline, and on average, coherence is equal to the left and to the right. And then once we enter these trial periods, then we have an average coherence which is different from 0. And that's what participants have to detect. But on top of this means coherence, there's a lot of noise in the actual coherence that participants are experiencing. And that means they cannot just see, oh, there was strong movement to the right just now, so I'll press the button, but they actually have to integrate what they're seeing over time to do well in this paradigm, because only if it's consistent across time for a few seconds that the average coherence is more to the left or more to the right, that would count as a trial. So we're forcing them to integrate evidence over time. And what this paradigm now allows us to do is we can look at the responses that participants emit. So for example, here I've marked some responses in green. So these were correct responses during a trial period. And we can look at the stimulus that led up to this response in all of the responses and average that across responses. And what we get is something like an integration kernel. And it tells us, the steeper this this kernel is, the more participants just went with what they had just seen. And the more flat this becomes, the more they integrated evidence over time. And maybe more interestingly, we can also do that with false alarms, so responses during baseline periods, where there wasn't actually any change in the mean signal that participants saw, but we can still do the same, use their responses to get this estimate of their integration kernel. And the other thing that this paradigm allows us to do when we record EEG data is we can make more efficient use of the data that we're recording, because in a conventional decision paradigm, we would only use a fraction of the EEG signal that we've been recording to actually estimate the neural signatures of decision formation, because there would be other parts of the paradigm, like intertrial intervals, which are non-informative, and so we would just basically discard this data. But in this new paradigm, we can use every sample of EEG data because participants are continuously engaging in this task and they're continuously integrating evidence, so they don't really get a break during these 5-minute blocks. And then we can use any interesting feature of the stimulus, how it changes over time, or even some output from our model, if we use one, as regressors to explain the changes in the EEG signal. And we've seen in data that this might allow us to estimate neural signatures of decision formation in much shorter amounts of time. So here we saw that we can get pretty good estimates of the neural response to changes in evidence in individual participants when we just use 10 minutes of their data to estimate this response, because the events are so densely packed, and we can really use every sample of the EEG to estimate our response. So the first study that was run with this paradigm was led by Maria Rissler, who was a PhD student at Lawrence Lab at that time, and she was also the one who developed the paradigm. And what she did is she varied across different conditions the frequency with which trial periods or signal changes would occur, and the length of these trial periods. And that was an attempt to kind of modulate how participants would integrate evidence over time, given different statistics of their environment, basically different change probabilities. And she found interesting reaction time differences and detection rate differences, etc., between these conditions. But I think most interestingly, the integration kernels differed such that in this environment where changes are more frequent, participants integrated over shorter timescales, so they had a more kind of high learning rate, if you will, in this integration process. And they did so over longer timescales when changes were more rare. So this is exactly what we've just been talking about in terms of expectations about the stability of the environment. How do we understand this computationally? So the perceptual decision-making process has traditionally been described using drift diffusion models, where a decision variable accumulates noisy evidence slowly over time until a decision threshold is reached and then a response is emitted. But we know that this perfect accumulation of evidence that is the pure DDM, pure drift diffusion model, is not optimal in our case because this paradigm places participants in a dynamic environment where things are changing. So you might not want to integrate over everything that you've seen, but rather you will want to put an emphasis on more recent pieces of evidence. And the way that we integrate this into the DDM is we introduce this leak parameter, which makes the integration process a leaky integration, or Ornstein-Uhlenbeck process, what's called. And the more leak you have, the more you tend to forget what you've previously seen and just focus on the most recent evidence. And we can apply this model to our paradigm and see that, so on top here again, you see the stimulus and the mean signal that underlies the stimulus. And at the bottom, you see some simulated OU process, so a leaky integration process. You see one example with a high leak, so more forgetting, and one with a low leak. And you can see that in if we've got a low leak, so we're more towards perfect accumulation and without any forgetting, then our decision variable is more stable. But it also means we're slower at detecting changes, so we might miss a few trials. Whereas if we use a high leak, then we lose some of the stability, and the decision variable might jump up with the evidence more quickly, so we might have more false alarms, but we're also detecting more of the trials. So it seems that there's this there's somewhat of a balance depending on the environment. And in fact, when Maria looked at the performance that different parameter sets, or different decision thresholds and different leak parameters result in, she saw that the optimal parameters did indeed differ across the different conditions in her paradigm. And the optimal model, which had different leak parameters across conditions, would make very similar predictions about the integration kernels that you could expect compared to what we see in actual human participants. Okay, let me also show you some of the EEG data. As I said, we can use this design to run a regression of the EEG signal using all kinds of interesting regressors. For example, the one that we're using here is the change in evidence at every time point of the paradigm. So on top, you see the stimulus and how it jumps around between different coherence levels. And then on the bottom, you see how we construct our regressor. And importantly, we're running this only during baseline periods, so where there isn't actually any change in mean signal. But it gives us an EEG response that shows us kind of how the brain reacts to jumps in evidence or changes in evidence. And Maria found that this signal actually differed significantly between the conditions, such that the signal was more sensitive to jumps in evidence during when signal changes were more rare. And she also ran a control experiment where she additionally introduced vertical motion in the stimulus, which followed exactly the same statistics as the horizontal motion, but it was completely irrelevant for the decision. So it was just sensory input. And she saw that the signal seems to be specific to the decision relevant information. So it's not just some sensory processing, but it seems to capture some decision related process. And I'll skip this one. But basically, again, what we're seeing is that our integration kernels might differ, depending on our expectations of how quickly things are changing. And let me just remind you, so the way that we propose this to happen is that if things are changing quickly, we might think we have less certainty in the current, in our belief about the current rule that underlies the stimuli that we're getting. And now we can extend this, what we've seen so far, in two directions. The first extension would be explicitly modeling the expectations that participants have. So I've showed you this OU process, and I told you that the optimal model in terms of best performance in this paradigm would show different leaks across different conditions. But in fact, there's now ways of modeling this directly in the evidence integration model, because obviously the DDM also derives from Bayesian belief updating equations. So we can put in these expectations about rate of change in the environment, and people have done that. And it turns out that this does exactly what we would expect. So it changes the leak parameter. And now something that would be really exciting would be to apply this to our data and be able to infer on kind of participant-specific expectations about stability and volatility. And I've been starting to do this here. So this is an example for such a nonlinear integration. And just an example for some parameter values that I've chosen. But basically what we can now do is we can vary the probability of transitioning into a trial or the probability of transitioning into a noise period, and see how that changes the inference process. The other extension that I want to mention is, so we've been talking about this precision of the current belief, but actually the learning rate also scales with the precision of the data. And so, you could think of a different set of expectations about how precise or noisy the signals that you're getting, which would have similar consequences for your integration kernels. And in fact, this is something that Leila, another PhD student from Lawrence, has tested or is testing already in an ongoing EEG study, where she's using the same paradigm, but she's varying the noise around the mean coherence signal. And we're currently still analyzing the data, so this is very preliminary. But what I've already seen in the first 11 participants when looking at their behavior is that they do adjust their integration strategy to these two different conditions. And note that now the way that I'm plotting these integration kernels is on the y-axis. I'm not only plotting average coherence, but now it's normalized by the standard deviation of that condition. And so you can see that participants seem to integrate evidence up to kind of one standard deviation away from the mean in both conditions, which basically means they adapt because the standard deviations are different in the two different conditions. And also when we fit exponential decay to these integration kernels, we see that the integration time constant is significantly higher for the high variance conditions, suggesting that participants are indeed integrating over longer time scales if there's more noise in the stimulus. The EEG data is still very noisy because we just have 11 participants, but there might be something in there too, which is nice. Yeah, so that's kind of what we've been doing so far. And now just maybe 2 or 3 minutes on what we're planning to do with this. So what we're currently hoping to test in a ketamine study, which will run hopefully this spring or summer, is using these continuous decision paradigms, which give us this reliable readout of participants' integration style and the corresponding EEG signatures. Will ketamine have the effect that I proposed in the beginning? Basically, will it decouple the integration process from these higher level expectations? And might that be an explanation for its psychotomimetic effects when we look at this in the perceptual domain? But then if we extend our continuous decision paradigms into other domains, can we maybe explain some of the other effects that ketamine has? For example, you could imagine that if ketamine decouples the processing of mood-relevant information, such as rewards or emotions, from higher level, maybe pessimistic expectations, that this might indeed be a good explanation for its very fast antidepressant effects. And again, kind of this is all an attempt to provide the missing link here and not look at the molecular effects of the drug, but rather look at the effects of the drug on certain algorithmic motifs and see whether these are similar across the different domains that the drug affects. To translate these continuous decision paradigms into other domains, we have a few ideas. And one of them is something that we're currently testing with Jan from EP. And this is a continuous reward decision paradigm where participants have to go fishing. And they have 4 different, or actually in the version that we're running now, it's just 3 different locations, but could be any number of different locations in which they can go fishing. And what they're seeing is in every location, they're seeing a few fish and a few algae, and the fish are rewarding and the algae are kind of punishing or losses. And they can sample different fishing locations using their eye movement. So whenever they look at a patch, they see the information. If they don't look at it directly, they don't see it. And then they can choose to harvest a certain location to get the rewards that are presented there by using button presses. And we hope that this design will, first of all, translate what we see in the perceptual domain into the reward domain, because the rate of reward and punishment will change dynamically over time. And so this is, again, a continuous paradigm where they can switch between the different patches whenever they want to, but they constantly have to observe the value of the different patches and compare them. But also what this hopefully allows us to do is to distinguish between decisions to harvest from a patch, kind of a value-based choice, versus decisions to sample from a patch using their eye movements. And note that these two are not in competition with each other. but basically you're sampling different patches in order to make a good decision about which patch to harvest. And what Jan and I are trying to do with this is something that I've been wanting to try for a while. So we've been hearing a lot about how high-level expectations about volatility or noise would shape the way that you integrate evidence. Now, something that I want to try is whether we can actually present the same stimulus across different conditions, but use different instructions that suggest that the stimulus might now be more noisy or more volatile, and see whether participants process the same stimulus differently using these different instructions. So this is an example for kind of the reward rate at 3 different patches, and the thick lines are the mean reward or the mean value of that patch over time, and the thin lines are the noise around that mean value. And you can see that there's kind of the value of the patch is rather, it's very stable across many seconds, tens of seconds in the mean, but it does change obviously much faster if you attend to the noise. And so what I'm now showing you is very exciting data. It's basically data for myself running this paradigm and trying as hard as I can to treat the stimulus differently across 2 different conditions. In the blue condition, I'm kind of assuming that everything is stable and the fluctuations are just noise. And in the red condition, I'm kind of going with the changes in the stimulus in the noise. And so, yeah, who knows, maybe we'll see something in actual participants as well, and not just when I'm doing this task. But I'd love to hear any feedback of what you think, whether people have done that, whether you know any effect like this. I think that's all I wanted to say. So thank you for your attention, and thanks to all of my lovely collaborators.
Thank you so much, Lilian. I think we should all briefly unmute ourselves and give a little clap. That was a fantastic talk. Doesn't work that well over Zoom, but you know it's meant. There's time for questions. I will stop the recording now.